Thank you. It was a fascinating talk. We are still 20 minutes behind time. Therefore, we will skip questions and we don't have time for this. And I'll call the next speaker. Next speaker is Professor Ian Owens, who is the head of the Department of Life Sciences in Imperial College. And he is also the director of science at the Natural History Museum in the UK. I am pleased. He will talk, by the way, I forgot. His topic is global patterns in biodiversity. Please.
Thank you very much. And like the other speakers, I'd like to thank the organisers, in particular Tamar, for bringing us here. It's been a really interesting few days, and it's really positive to feel the energy that's in this area at the moment in terms of developing a biodiversity programme and so on. I was particularly impressed actually to see that large hole in the ground that there is a few hundred metres over there, which Tamar promises me is the beginning of a new museum. So obviously from my background, it will be great to be able to welcome Israel into all the collaborations that go on in that area. So very exciting. Today, I'm going to try and get you interested in the question at the top of this screen, which is the question about what determines large-scale patterns in biodiversity. I should warn you now that I'm not going to answer that question, not because it's a secret, but because I don't know the answer. And I think that's one of the reasons why this is such an interesting area at the moment. There are some genuinely large questions which have strong relevance to society, which we simply don't know the answer to yet. And we need to know the answer to them. So rather than to get you interested in that question without being able to answer it, I'm going to first of all tell you a little bit, 3 sections of the talk you can see there. I'm going to tell you a little bit about, I think, broadly what we used to think we knew about large-scale questions in biodiversity based on limited data. Then I'll tell you a little, give you a flavour of the cutting edge of this sort of field at the moment, talk about a bit of work that we've been doing in our lab over the last few years. But probably most important at the end there, I'll try and emphasize some areas that I think would be really exciting to get into. And I think that given Israel's strength in lots of areas of science and mathematics, it's the sort of area where Israel could have a big impact. So first of all, what did we used to think we know about large-scale patterns in biodiversity? Well, it's almost embarrassing now to look back to say that there's a whole series of textbooks that have been written. You could go to the library here and get them. Up until just a few years ago, most of what we would say about large-scale patterns were based on a couple of regions, well, you can barely call them regions of the globe. An awful lot of big ideas about biodiversity come from one small, probably not totally typical country from the UK, simply because the UK is rather overcrowded. There's not much to do there, so people go out and watch birds. The one good thing about that is you do have lots of information on a few groups like birds and like butterflies that go back quite a long time. So you can start to analyse biodiversity patterns. It's a hard stretch, though, to say they're really large-scale biodiversity patterns. As somebody cruelly put it to me the other day, it's a small, cold island off the shore of France. Which even I have noticed that in the last few years. The other place where there's been large ecological data sets, of course, is from North America, and particularly from the US. Again, I haven't been to the US all that often, but I guess it's cold and wet and so on, so they spend a lot of time watching birds from what I can see. So there's lots of good data from the US. It's a near continental scale. But as I'm going to show you, hopefully illustrate in the coming minutes, I'm not sure that even if we did understand biogeographic patterns or biological patterns in the UK and the US, would that necessarily tell us, or has that told us, how other parts of the world work? Particularly those areas of the world in which we're probably most interested, where there's really a lot of biodiversity left. So what's changed in how we've attacked questions in biodiversity over the last few years? Well, as several people this afternoon will illustrate, a lot of it has been determined by getting together really big data sets. This is a data set that some colleagues and myself, based on many decades of work by field biologists all over the world, we pulled together a database of the breeding distribution of birds in the world. So you'll recognize many of the places that people have already talked about and many of the classic hotspot areas. You can see, for example, the Andes here, various bits of the Amazon basin, the Atlantic forests, various bits of the rift, the mountains associated with the Rift Valley, the slopes of the Himalayas, and the mountains of southern China. Similar databases now exist for other groups, such as for amphibians, for mammals, for some groups of plants and so on. What this has allowed us to do is to ask whether the patterns that we previously found for little areas like the UK and the US, whether the patterns we discovered there also applied on this broader scale. And what I'm going to contend broadly is that they don't fit very well. There's a lot more for us to understand once we start looking at this global perspective. Here are a couple of examples of that. Firstly, there's been a lot of discussion about whether this distribution, the extent to which it can be explained by neutral mechanisms alone. Because if you think about it, there I pointed some of the high spots in terms of avian diversity. You can see in dark blue some of the cold spots for that index. But one of the questions has been, what would you expect if species were just randomly distributed across that terrestrial surface? You wouldn't expect it to be just a perfect yellow color all the way across. You'd expect some heterogeneity. So is there more heterogeneity there than expected by chance? When people looked within particular regions, particularly within sub-Saharan Africa, for example, people thought that possibly the pattern within sub-Saharan Africa could be explained to a reasonable extent by neutral mechanisms. So what you can do with these larger databases is that you can apply those sorts of neutral models across, in this case, the entire terrestrial surface. So this slide at the top there, you can see this is the observed distribution of birds. This is what happens if you take the birds off that top map, you take each one, you imagine, you take its geographical range, and then you throw that range on here randomly. And the way that you do that is you take the first square of that range, throw it on there, it hits a random spot, and then you allow it to spread out like ink spreading across the surface. Now you can see if you do that, so it's a relatively biological neutral model, if you like. You can see that if you do that, the model that you get here, the overall neutral model, doesn't look very much like the observed distribution at all. You can also demonstrate that it's not a very good fit by doing things like, for example, plotting this is the observed species richness. So this is looking at each about 100 kilometer by 100 kilometer cell across the terrestrial surface. This is the observed distribution, so how many species we know are there, and how many species are predicted to be there by a fully neutral model. You can see that even with somebody from a behavioral ecological background, that's not a terribly strong relationship. You can see that it doesn't even extend far enough along this axis to explain the great peaks in diversity that you see. Another thing that you can do is you can look at that bottom graph there and look at biogeographical rules that we think exist, such as the latitudinal species gradient. What this graph here on the right shows is the pale grey bar shows the observed latitudinal gradient in species richness. So this is going from, in the middle there, that's the equator, over to the right, that's extreme northern latitudes, and down here, extreme southern latitudes. So we'll talk about this in a few moments' time, but this is the observed latitudinal pattern of avian species richness. This is the pattern you get from the fully random model. You can see it doesn't pick up the richness that's there. It's a very even model. However, we haven't just got more data these days. We've also got better maths. We can do better neutral models, if you like. And we've also got almost limitless computational power. It's always slightly dangerous to say that because, of course, we can always think up fancier and fancier models that need more and more computers. But for some of these sorts of analyses, the amount of computational power that we have now is no longer the limiting factor. So one thing we can do is, instead of just doing normal neutral models, where we, for example, pick a species off this distribution map and just throw it randomly onto the terrestrial surface, instead we can weight that terrestrial surface. So we can weight it by things such as energy, for example, which is what this graph shows here. So that the species are more likely to go into those areas where there's high energy, in this case evapotranspiration. Or you could weight it to do with altitude, or you could weight it to do with temperature range, or whatever environmental variables you think might be important. The idea there is that if you pick the right variables and you throw the species on there, what you get, the nearly neutral model, if you like, should be more like reality. Whereas if the variable that you picked isn't associated with the real patterns, or isn't correlated with the real patterns, it shouldn't make a meaningful difference. Now you can do these in all sorts of complex ways, I'll show you in a minute, but here's the most simple and the most effective single variable. If instead of throwing species completely randomly at this terrestrial surface, you just weight both where they go and their spread by energy values, so environmental energy values, as I said, in this case evapotranspiration, this is the sort of pattern that you get, which looks quite different, if you remember, from the full neutral model here. It looks quite a lot more like the observed model, which is on the top left here. And we can test that by, here is again the same graph of observed values versus predicted values, a much stronger relationship. A great deal of scatter, still a lot to be explained, but at least a strong relationship. Also, you can then plot the latitudinal trend for the observed data versus the predicted data. And as you can see from the right there, there's a much stronger relationship amongst those. Now, of course, what you can do is you can weight the terrestrial surface in all sorts of different ways and add lots of different variables in there and do the equivalent, if you like, of a stepwise model where you decide which of these variables helps you to predict where the species are. There's an enormous variety of ways in which you can do that, and different scales and different neutral models and so on. But when you do that, you come down to a pretty predictable set of variables. What it basically comes down to is you get strong correlations with high altitude, and particularly altitudinal range, and to do with the amount of energy that's available. What these MDE numbers at the bottom here show is that this is a particular form of neutral model, and this is suggesting that in addition to having temperature and altitude, there are also some effects of the shape of the continents. So you tend to get greater species richness near the middle of continents, where it's easier to pack in more species. Thanks. Okay, so using these new databases, these new forms of analyses, and this more computational power, you can do richer and richer analyses of things like species richness. But you can also revisit some of the old biogeographical rules that we thought existed. One of the ones I mentioned a few minutes ago was the famous latitudinal species richness gradient. Ecologists such as Robert Ricklefs have suggested that the fact that we still don't understand why there are more species at the tropics mocks our understanding of ecology. But what happens when we look in more detail using these large databases? What happens when we look at latitudinal gradients? Well, I showed you it before, but let's have a look in more detail now. Here is that gradient again. Equator in the middle, North Pole up that end, South Pole down this end. Now, if you do fancier and fancier analyses, what you're expecting here is to get a peak in the middle and it going off towards the edges, towards the poles. That's what you'd certainly expect if you looked at North America. Well, what those dark circles there show is what the median species richness is for each of these latitudinal bands. And hopefully you can see, if you just look at the dark line, you can see that there is a tendency for a peak towards the center. If you do various forms of analysis, you can demonstrate that's a significant tendency. But hopefully, if you look at it a bit longer and you look at the little grey dots, you can see there's an enormous amount of variation around that line. And I think if you were the first person looking at this distribution, you would not only notice the central tendency, you'd also notice there's a lot of variation to be explained. You can probably figure out if where you put Israel on that line, over towards the northern temperate region there, you can see that there's a particularly large amount of variation in that zone. And that's probably because of the bringing together of some wet areas and some major desert areas. I would say that those combinations, particularly the desert areas, are one of the major biological phenomena we don't understand at the moment. And you'll see that band of latitudes come out again and again on these sorts of analyses. So more data suggests that the old patterns are statistically there, but they don't really describe the full richness of the situation. And the same is true for some other phenomena as well. Here are another couple of rules that are often brought out from in classic biogeographical textbooks, in this case based on North American data usually. On the left here we have the relationship between geographical range size on the vertical axis and latitude on the horizontal axis. And what this is showing is that as you go away from the equator, away from the temperate zone, towards the polar regions, geographical ranges tend to get bigger. This is what's called Rappaport's rule. And it's potentially very important because, of course, it's one of the potential explanations for why you get less species at high latitudes and more species near the tropics. It may simply be that if you need a greater latitudinal range towards the polar regions, it's hard to pack in a lot of species there. Whereas if species can exist with smaller ranges near the tropics, that may be one of the ways in which you pack in lots of species in those areas. But of course that presumes that this rule is general across areas outside North America. Another famous rule is that called Bergmann's rule. This is again a latitudinal or proposed latitudinal trend with respect to body size. In this case, the surrogate of latitude that's being used here is annual mean temperature for different regions. In this case, going from low temperatures to high temperatures, so scaling the other way. So you've got polar regions towards this end of the horizontal axis and tropical regions towards the far end of the horizontal axis. And then in this case, body weight of different human groups on the vertical axis. And you can see that people tend to be larger towards polar regions. And this is a pattern that's been thought to exist in not just mammals, but also various other groups, particularly of warm-blooded creatures. So Rappaport's rule and Bergmann's rule are probably two of the top half-dozen generally thought to be true biogeographical rules. The trouble is that when people have tried to analyze these in other contexts, using either other groups or different regions or things like that, there's been very variable support for these sorts of rules, which either suggests that the other analyses aren't as good, or else there's some heterogeneity going on across different areas of the world. Well, of course, with these big databases, you can now test this. You can apply roughly the same analytical techniques and use the same variables across the whole globe. And here's what happens when you do that. So again, we've got analyses of geographical range size on the left here, and body size on the right there. On the top, those are the maps of those traits. So that's a map of geographical range size. So the areas that are shaded red are those areas where the species present tend to have really large geographical ranges. The areas colored blue tend to be those areas where you've got tiny little geographical ranges, so typically endemic species. With respect to body size there, the areas that have got deep brown have got large-bodied organisms, where the ones which are blue have got small-bodied organisms. This is again, all bird data. Now let's look at those latitudinal trends. Well, remember on this graph here, what we're expecting is a U-shaped curve. Because from North America, we know that, or for the groups that have been studied in North America, we know that species tend to have larger geographical ranges towards the north and smaller geographical ranges as you go down towards Mexico. And you can see on that graph there, you get the beginnings of that relationship. However, what happens in the southern hemisphere? Instead of this relationship being reflected, or a mirror reflection in the southern hemisphere, you actually get the opposite. You actually get geographical ranges getting smaller and smaller as you go into that southern hemisphere. So you don't get the U-shaped relationship that you'd expect just from the North American data. You're already probably beginning to suspect why North America gives you different patterns from other parts of the world. You can also see here, again, there's a great deal of scatter in these relationships. You can demonstrate that this is a significant trend, but there's all sorts of sub-trends in there as well. You can see, for example, here, there's a big cloud of data points here and a big cloud of data points here. These are effectively two different biogeographical regions which have differences in the relationship between geographical range size and latitude. At the moment, we don't understand the explanation for that. When you look at the data for body size, it's a little different. Here, we're again expecting a U-shaped relationship. And again, you can see you can detect that U-shaped relationship. In this case, it's actually quite difficult to demonstrate that it's statistically significant. But there is a relationship there. Here you can see there's a great deal of scatter. And again, you can see these large bands appearing north and south of the equator, where you get enormous variation in body size. So if you were to ask, does Bergmann's rule exist? You'd be able to say, well, statistically it does exist, just. However, when you look at it in more detail, you find that Bergmann's rule is actually underwritten by a whole series of other rules, which makes you ask whether the physiological phenomena that Bergmann based his rule on are actually the ones that determine this. And again, you can see that just in the northern hemisphere, that enormous amount of variation in body size. That's driven by the similar latitudes, a belt of land which is a combination of some near-tropical, wet regions, and a great big arid zone. Again, we have very little understanding of the relationship there between temperature, aridity, and other ecological factors. I'll miss out this slide in any detail. All this slide shows you is this is an expansion of the body size analysis. On the left here, what this shows is, rather than just fitting temperature, if you try and fit a whole series of other environmental variables, do you get significant relationships? Yes, you get a whole series of these sorts of relationships. And you can see that quite a lot of them aren't linear, which again is something that you couldn't extract from the North American data alone. What the graphs on the right-hand side there are saying are asking, do these differences occur within species, such as within genera, such as we illustrated with the human data there? Are these things that happen? So is the reason that there are, for example, large-bodied things in the polar regions, is that because all of the creatures that are there, that have similar species elsewhere, is it because they've all got bigger? Or is it actually because you only get large-bodied forms there, you only get owls and wolves or whatever else it is there? Again, it turns out to be a combination of those sorts of rules. Some of the variation in body size turns out to be variation within groups, but quite a lot of it also turns out to be sorting amongst different sorts of groups. So again and again you get, with the more data that you get richer examples, and you find that many of the rules that you discovered in the UK and in the US aren't general when you go out into other arguably more interesting biodiversity areas. So what can you do about this? What would I say the future of this sort of work was, this particular area of work? Well, one avenue that we've been exploring is trying to bring together not just the spatial analyses that I've tended to focus on there, but bringing together a combination of spatial analyses and phylogenetic analyses. Because remember when I was talking at the beginning about how we build those neutral models and so on, I talked about getting species, throwing them onto a terrestrial land surface and letting them spread out. That's a relatively sophisticated way of doing spatial models, but actually we know what happens in these sorts of systems is you're not throwing new species on there. Those species are speciating from each other, diversifying, going extinct, and so on. So with these sorts of models, you should really be trying to grow your own garden, if you like, of biodiversity, combining not just spatial patterns, but also the phylogenetic branching pattern. Now, people in the past have suggested this sort of thing. Here are a couple of graphs out of a paper that I really admire from about 20 years ago. This was a paper which essentially said, at the moment, this is 20 years ago, most of what we think we know about deep evolutionary patterns we try and get from fossil material or its equivalent. What these guys were saying is that perhaps that's not the only source of information about deep history. Perhaps phylogenies based on living species, particularly molecular phylogenies based on living species, can also tell us things that happened a very long time ago. And a couple of phenomena that these people picked up on to try and analyze were, on the left here, what's called the rate of divergence. So how quickly, so this is a little phylogeny here. You can see that this is imagining an original species, speciating, some lineages going extinct and so on, and here are the extant species that we could sample today for a normal molecular phylogeny. What they were trying to figure out is, does this branching pattern, particularly when you plot the number of branches against time, does this tell us about the amount of speciation or the amount of extinction that was going on here? And one of the main things that came out of this is, how random are these patterns? Again, it comes down again and again to being able to figure out whether what you're seeing is random or whether there's something unusual going on there. So people became interested in whether these phylogenetic trees are more unbalanced than you'd expect by chance alone. So in this case, people are interested in whether you get these sorts of patterns, so you get an initial apparent burst of phylogenetic divergence, and then what's called a slowdown after that. The other thing that they were interested in, which I mentioned by mistake then, is how balanced these trees are. What this graph here shows you is, this is a histogram of how many species there are in a whole series of avian families. So imagine you line up all the avian families, 100 or so families, count how many species there are in each of those, and draw a histogram of those species counts. So these are families that have got loads and loads of species in them. These are families which have got very few. And this is the number of cases. Now, what you can see from this graph is that most lineages, or most families, contain rather few living taxa. There's lots of cases over on the left here. A few of them contain way more taxa. In fact, they contain thousands of taxa if you do species within families. Now, initially, this looks like a non-random distribution. So the bars there show you the real data. The little black line shows you a particular neutral model. It appears that there are too many families which have got too many species in them. And what this led people to start to ask is, what makes the difference between a family being represented by a single living organism, such as an ostrich or something like that, versus other families which are represented sometimes by thousands of living examples? Now, lots and lots of different groups, including ourselves, have come up with all sorts of adaptive explanations for this. This is just illustrating there are better and better molecular phylogenies available these days. This is for a group of passerine birds. And lots of groups have tried to use these phylogenies to try and figure out which ecological variables might explain the differences in tree shape. However, typically people have only done that either from a phylogenetic angle, such as in this case, or from a spatial angle. What we've been doing over the last few years is building a modeling framework in which you can combine both spatial and phylogenetic components. So here's a simplification of this. You start off, for example, so the square block there in A, the outside surface is the entire universe that you're interested in. That is the original species, if you like, shaded in. Now you assume that this speciates, shown in B. There can be range shifts and another species coming in. And gradually, with extinction and speciation and range shifts, you can end up with that's that pretend garden of biodiversity. Now this, if you like, this is an emulation. Well, this is a neutral model of spatial diversity. But of course, because you've allowed this model to evolve from a single original species through all of these processes, you also know which of these species is related to which other species. So as well as showing the spatial pattern there, you can also draw the phylogenetic pattern upon which they were based. That means that when you then compare these sorts of models with real data, you can figure out, you can handle both the geographical components of it and the phylogenetic components of it. So, of course, we've applied these sorts of techniques to those two phenomena that I was talking about before. Shifts in the rate of divergence, so slowdown in speciation, and also how many species how many species there are within particular lineages. So this axis here shows you whether there's shifts in the rate of diversification over time. So anything positive here suggests that speciation is getting faster. Anything slow suggests that speciation is getting slower. Here's the observed data for birds. It's slightly below that 0 line, and this is often interpreted as a slowdown in the rate of speciation in birds. You can see there's a lot of variation around that, depending which particular group you're looking at, and this has led to controversy. Is there really a slowdown in birds, or is there just a lot of variation in what's going on in birds? Well, when you just use a simple phylogenetic model to try and analyze this, without any space involved, these are the sorts of neutral predictions you get. Now you can demonstrate, so this is just phylogenetic neutrality. Using different sorts of tests, you can show that this observed data is actually different from these models, or for some of these models, so you can come to the conclusion birds are slowing down in the rate at which they're speciating. However, what happens, what I'm going to show you over here, is what happens when you don't just have phylogenetic neutrality, you also know about spatial neutrality at all. What that shows you is that this is a whole series of new models that also bring in spatial variation. What this demonstrates is that if you bring in space, you can create almost any pattern that you want. Now, in one way, that's depressing, because it shows that this observed variation in the rate of speciation amongst birds is potentially consistent with all sorts of neutral models, which is depressing if you're wanting to find the one magical trait that makes things speciate. But it may also be accurate. It may also tell you that this sort of information alone doesn't really allow you to discern amongst major biodiversity creating processes. Briefly, the same sort of thing happens if you look at tree imbalance, phylogenetic tree imbalance as well. If you look at just the observed data here, it looks as though there's a slight negative trend. With different sorts of phylogenetic model, you can sometimes demonstrate there's a slight perturbation away from that. You can just about detect a significant difference. However, once you add space to the models, then you can get all sorts of patterns in there as well. You can use the same sort of thing to look at. These are some long-term trends that have been shown in different groups. For example, on the left here, this is what tends to happen if you get hold of some fossils and show how the geographical range of species change over time. What you tend to get is you tend to get an initial rise in the geographical range of species, and then a fall in that range, and the species goes extinct. And this is what's being called the life and death of a species. And based on the fossil evidence, this appears to be quite common. This really has been found in lots of different groups. And it's thought that maybe you can predict what happens to a species over time through these rise and fall sorts of models. On the other hand, when you look at some living groups, for example, if you use a molecular phylogeny, look within birds, and try and plot the age of a species on the horizontal axis versus the range size of different species, for living species, you typically don't get that. You only get the beginning of that curve, or it appears like just the beginning of that curve. You just get species having bigger geographical ranges. What we began to wonder is this actually telling you something about the birth and death of species? Or could these patterns also be random? So what we did is we got a whole load of species. We put them into our framework. We allowed them to be born, to go extinct, to grow, to change, and so on over time. Now, when you do that, you get, it looks a bit like Brownian motion there. So this is phylogenetic and spatial Brownian motion, if you like. You get all sorts of variation in there. Now, these are just neutral models with different lineages, different species, if you like, showing all of their different trends. What happens now if you pick from this model, from this neutral family of species, if you pick all of the ones which went extinct and show those at the bottom versus all those that didn't go extinct and show those at the top? Now, if that doesn't make any difference, you should see roughly that graph shown at the bottom and at the top. Instead, what you find are these sorts of patterns. If you pick just neutral species and just select the ones that happen to go extinct during your simulation and draw those, you get the old pattern of apparent species birth and death. If, on the other hand, you just focus on those species that survive through the simulation, you get the other pattern of apparent growth in geographical ranges. Now, that's nothing to do with differences amongst these species. They're all just completely neutral species. This is just a sampling artifact of what happens when you sample this sort of set of Brownian motions for a particular trait, for a particular characteristic. So when you go back to these old patterns here, it's actually, if you then compare those sorts of simulations with this real data, you can't detect any difference from neutrality. Which again, is initially, I admit, it's initially depressing that from these very old patterns you can't extract the biological information so easily that we thought. But my guess is it might actually clear up some issues with respect to differences amongst databases and so on. Okay, I'll stop talking now about the things that we've been doing and talk about things that I think other people should do, which is always more fun. I mentioned before that almost everything I've just shown you, with a few exceptions for fossils and so on, have almost all been based on living bird data. Now, to be fair, there's data available on more than birds. So here, for example, is data on birds, mammals, and amphibians. Here, split across all species, endemic or rare species, and species which are listed as being threatened by extinction. But if you try to expand this out and look at the groups that you might possibly be interested in, such as lots of groups of microbes or plants or invertebrates and so on, the same sort of data just isn't there. So in the same way as I've spent quite a lot of this talk saying, getting this sort of data has shown us why the old data just on the UK and the US wasn't telling us about the whole picture. What I'm now concerned about is, even with this sort of data, is it telling us about what's happening with invertebrates, with marine groups, and with microbes, and so on? And what I'm fearful of at the moment is that we just don't know enough about even how many species there are in those other groups in order to make those sorts of estimations. At the risk of annoying Stuart Pym, which is relatively easily done, I would say, but at the risk of doing it anyway, I'm going to show a different set of analyses about trying to estimate how many unknown species there might be out there. Many of you have probably heard of this debate about how many species there might be. Well, one approach, but probably not the only approach, is to try and estimate how many species there are out there by looking at how many species we discovered over time and what the shape of that curve is. So if you like, trying to predict where the asymptote of species discovery curves might be. Now, Stuart will talk to you about problems with that approach this afternoon, but for now I'm going to look at the sorts of relationship that some other groups have found. So here are species discovery curves across phyla, classes, order, for a whole series of different groups, which you can see over on the left here. What these groups have said is that they think that for quite a few of these curves they can detect an asymptotic effect, and they've tried to estimate where that might be. By doing that for different phylogenetic levels, they can try and come up with an overall estimate of not just how many species there might be in total, but also where those missing species might be. And here's a table rather that summarizes that data. So you can see different taxonomic groups here. You can see that how many of them have currently been described. And you can see the numbers which are predicted to occur from analyses of these species discovery curves. Now, I don't expect you to go through this in great detail. The first thing that I'd like you to note is that the estimate is that there should be at least 8 million species out there. So we've only described a fraction of them. This is just amongst eukaryotic organisms. So there's probably a vast number of undescribed species out there, particularly in some groups. Also look at the number that are estimated to be in marine environments. Everything that I've just spoken about, almost everything that people typically talk about in biodiversity circles, are based on terrestrial organisms. These sorts of models predict there's a vast amount of unexplored marine diversity. And lastly, of course, look at how few prokaryotes are on this analysis and how few prokaryotes are estimated to exist. Even as a bird biologist, I would venture that's probably not an accurate reflection of how many prokaryotes are out there. What it is I think, a reflection that we don't even have enough baseline data to even begin to estimate how many prokaryotes we don't yet know about. And that isn't just all because we don't know how to estimate what's a species in some of those groups. We have methods to get around that a little bit. We're just ignorant of vast parts of our biodiversity, which again, in some ways is depressing, but I find enormously stimulating because there's so much out there to go and explore. Now, coming back to a few things I said in my introductory remarks, how are we going to find these species? Well, I think one approach is to make use of the collections that we already have. In the collections around the world, we have many millions of different creatures, some of which are described, some of which aren't. Some of which are being collected in rather bizarre ways, but some of which are being collected in reasonably structured ways. My guess is that in order to start to estimate, to do some of the biodiversity estimates, what we have currently locked away in museums is a valuable start. Particularly when you start to bring together the information that's in different international museums. This slide here is a snapshot from a few days ago. Each of those yellow dots on there, going into the red dots, is showing you a sample from a museum somewhere in the world that's currently loaded onto GBIF. That's about 180 million data points of organisms by their geolocation, probably with some pretty good taxonomy associated with them if they're in GBIF. Now, it's only a start. This is only a small fraction of the amount of data that's available in the world around museums. Now, it's clearly very messy data. This is hugely biased with respect to the usual regions that you'd expect. But everything that you've been looking at over the last half hour started out as very messy data. Ecologists aren't bad at dealing with a bit of mess in their data. And I think this is a treasure trove for us to start mining to get early information on other aspects of biodiversity. Okay, I'll leave it there. The only thing I wanted you to look at on this last slide is a few of these boxes here. This is really me trying to convince you that the sorts of approaches that we now have available, that I suspect will come up with lots of the speakers, which are a combination of more data, particularly more genetic information, new analytical tools, and new computational tools, mean that there's a new, if you like, technical revolution in biodiversity studies, I think, which again, I would say that the Israeli scientific community is very well placed to exploit. Thank you very much.